Well, thank you for braving the rain to be here. I appreciate it. Hopefully teachers give you extra credit for putting that effort forward. So let me start with my journey to STEM, because that's one of the bullet points that need to be covered. So I was born and raised in Bakersfield, California. For those of you who aren't familiar, that's in the southern end of the San Joaquin Valley, or the Central Valley. It's going to be a pretty large city. It's now just about 500,000 folks in the city itself. But essentially it's still an ag and oil town. So surrounding the city, lots and lots of agricultural land. The way my kind of family was oriented, I wasn't directly involved in agriculture, like my parents weren't farmers or anything, but I was always sort of around it. Certainly some of my family members would work in the fields. Back then you actually still manually pick cotton in the southern San Joaquin. There was a certain amount of involvement in the UFW, Cesar Chavez and Dolores Huerta back then. So I was kind of around the periphery, if you will, of agriculture in that way. And then also, growing up as more or less a suburban kid, I had lots of buddies, lots of friends who grew up on farms, right? Dairymen and folks who had orchards. So kind of on both sides of that spectrum, I definitely had kind of agriculture in the background. So I went to high school in Bakersfield, went to Bakersfield College in Bakersfield, so I got the community college education that you guys are getting right now. And that set me up pretty nicely to not know exactly what I wanted to do, but I had a good sense that I wanted to be outdoors. I wanted to do something that made a difference, as tragic as that sounds. I wanted to do something that was a little bit bigger than myself, and probably something that had to do with some kind of environmental science. I didn't exactly know what. So I went to San Jose State and got into the environmental studies department, and I felt like it was a major accomplishment just determining a major, just sort of getting to that point. I felt like I had accomplished a lot. So I got to the ES department, and the first thing they said is, all right, congrats, welcome, now you need to pick an emphasis. You need to pick an area of concentration so that you can begin to specialize and focus on a particular course of classes. And I immediately felt discouraged again, because now I had to start all over to figure out what the heck I wanted to do. The notion of sustainable agriculture, the notion of organic agriculture, the notion of working with a different model for growing food really became appealing to me. Again, growing up, I was always around agriculture. It seemed very interesting, it seemed very compelling for lots of different reasons, but I never could kind of get an intellectual or emotional foothold into that particular setting. But when you begin to change the theoretical paradigm for how food is grown, all of a sudden then it became kind of interesting, kind of exciting. All right, now I can sort of see how you can arrange the puzzle pieces and maybe, kind of get in there and do some interesting work. And I learned a lot of stuff about agriculture, sustainable and not, right? Certainly the context for wanting to improve things are pretty obvious. We apply a lot of pesticides in California, a heck of a lot. This is California Department of Pesticide Regulation data. 173 million pounds of active ingredient pesticides are applied in California alone. If you look at, this is CDC work, the number of pesticides that come up per person on average through blood work and urine tests. The most common was 13 different pesticides or pesticide breakdown products are found in us, right? So that's certainly might be a cause for concern. Similar work, there are urine tests and blood tests show the percentage of people who were sampled and the different chemicals that might show up in our bodies. Anyone know what DDE is with the red arrow? That's a breakdown product of DDT. You may or may not be familiar with that, right? That was kind of the OG of insecticides. That was first started being used in the 40s and the 50s. It was very successful, and with the help of Rachel Carson and others, we started to figure out this is not very good for the environment. Any guesses as to when it was banned? In the US at least. Any guesses? Yeah, 72. It It was banned in '72. This work was done around 2000. So these chemicals, once they're applied, they don't go away very easily. Some of them have an incredible ability to persist in the environment. And when you look at pesticide exposure and health, there are so many studies that demonstrate at least a corollary relationship between pesticide exposure and a lot of ills. It could be miscarriages, it could be cancer, it could be Parkinson's disease. There are lots and lots and lots. The epidemiology-based cause and effect science isn't there yet, but we know what's being applied. We know that a lot of what's being applied are, have very high carcinogenic rates or mutagenic rates. We know that they get off farms through drift, through runoff. We know that farm workers are exposed. And we know that, at least in a correlation type fashion, right, what's being applied makes you more likely to experience some of these chronic diseases. So the context for me that integrated both the environment, farm worker safety, rural communities, and consumers, right, because we eat this stuff too, was pretty profound. That was definitely compelling. So now the sort of idea was, okay, what do I do with this? How do I kind of pivot from an interest to action? So I was in a class at San Jose State, and there was a researcher from UC Santa Cruz who was giving a talk about the research they were doing in organic cotton in the Central Valley. I pretty much jumped out of my chair, grabbed this person and said, please, please, please let me intern with the research office. I really need to do this. This sounds perfect. And what it was really cutting edge research, looking at the time, what was a burgeoning crop, which was organic cotton, over by Chowchilla, Merced, that area. And it did a full inventory of the insect community and a lot of the growing indices, yield, and everything else of organic cotton, of IPM cotton, that's integrated pest management cotton, in other words, sort of low spread, and fully conventional or high spread. And it was a great opportunity for me to really get the basics of entomology and how entomology works in agriculture. So you can see that here's the guy with the sweep net, right? You get insect counts just again and again and again. There was nothing necessarily sophisticated about the work, but it was comprehensive, and it was a wonderful introduction to insect communities, some of the profound changes that insect communities undergo in different growing environments, and the culture of agriculture, right, which is its own particular thing. So one of the things I love about entomology and agriculture is there are clear rooting sides. You have good guys and bad guys, or good bugs and bad bugs, right? And the whole notion of pest management or sustainable pest management is you want to tip the scale so that the good bugs have a better likelihood of succeeding than the bad bugs. So here on the left, we have a syrphid fly or a hoverfly. This is a fly that mimics a bee, and it lays eggs that hatch out into predaceous larvae. Do you guys know the name of a fly larvae? What's the common name of a fly larvae? A maggot, that's right. So here we have predaceous maggots. And so there's this really small maggot that ends up being the most important biological control agent to kill aphids on the coast for vegetables, for broccoli and lettuce and spinach. They all rely on these really small maggots that eat hundreds and hundreds and hundreds of aphids. So there are all kinds of interesting research endeavors to try and figure out, okay, how do we best set up ecological designs in vegetables on the coast so that these little maggots can eat as many aphids as possible. And then the aphids don't contaminate our food, right? So I just loved the idea, and continue to this day to love the notion that there are little mini battles going on in every single field, right? Every single week there's a little battle that's happening. And you've got to figure out how to win this war every single season. It's fun, it's dynamic, it's hands-on. And just to this day, I really, really enjoy it. And then you can take a step back and look at an applied science like this, entomology, and we're not working in a vacuum, right? We're not looking at predator-prey relationships in a redwood forest that's relatively undisturbed. We're looking at applied science in a very, very big system. And there are forces that are at play that are much bigger than us. There are economic systems, cultural systems, and then biological systems. And all of these massive forces, right, can have a huge influence on the viability of a project. You might have a wonderful theoretical approach that's based in really great ecology for success, but if it doesn't improve the yields of a crop, it's not gonna work. If it doesn't adhere to the culture of that particular segment of agriculture, it's not gonna work. And if it's not consistent with these broader themes and characteristics of a particular ecosystem, it's not going to work. So it's this highly applied system, and you've got to find the sweet spot for what is going to be successful. It's constantly changing. It's just tremendously interesting. I'll give you a quick example. There's a new pest in strawberry. It's an exotic pest, and it gets into the fruit. It gets into strawberry. And a way to delay and mitigate this infestation is to have workers come and kind of pick the fruit off the plant and remove it from the field, right? Because you're stopping that next generation from being able to reproduce. Labor, I don't know if you've heard, farmers are having a really hard time finding enough labor to get anything done, particularly to pick the crops, because of some of these larger, geopolitical things that are happening in the Western Hemisphere. So there isn't enough labor to sort of tackle this relatively, at least on paper, would be a simple task to reduce this pest pressure. So because of what's happening on the labor side, it's exacerbating the pressure that this pest is exerting in strawberry. And that's just one of a gazillion examples of how, when you're working in this larger system, there's so many things that have to be taken into account. All right, speaking of strawberry, I thought I'd go over some of the research I've done pretty quickly. I should also, I guess, take a step back and say, so I took that internship with UC Santa Cruz in cotton, and I just was persistent. I just hung around and stuck around and tried to be as proactive as possible. I tried to be as helpful as possible. I tried to ask as many questions as possible. Volunteered for everything, was just super excited, and that wasn't a front. I was genuinely really excited about the work, and I never left. Basically, I got hired on as a lab assistant 1, which is basically paid grunt work, and I just kept on staying and sticking around and climbing up the ladder. I got hired in 2001, and I've been there just ever since, really enjoying the work. Okay, so with that said, I received my master's. There's sort of two fronts here, right? My educational background and then the research itself. I'll get my background out of the way here real quick. I received my master's at San Jose State as well while I worked at UC Santa Cruz. So I was able to utilize the resources and faculty from both campuses at the same time. If you have a chance to do that, I highly recommend it. It turns out to be really rewarding and really useful. It's a good way to go. Okay, so on to strawberries. You may or may not be aware California is sort of the hub of strawberry production in the country, right? We produce over 90% of the berries in the country. The percent of those berries that are organic increases every single year. It just surpassed 10% this year, at least in terms of acreage. That's 10% of the berries that are grown in California. The percentage of organic fruit, the percentage of organic strawberries in California is very high. Okay, another thing I love about insects is they're super cool looking. It might take a while to kind of get past the, a little bit of the ick factor, but once you do, they're really neat. So this is Lygus hesperus. This is the Western tarnished plant bug, or Lygus bug for short. And this is an adult. And it's the primary pest in strawberry. Here's a side view of the same adult. It's polyphagous. Do you guys know what that means? Polyphagous, polyphagous. What does poly mean? Many. That's right. So it can eat many different kinds of plants. It's a generalist. It can eat over 200. It can feed on over 200 different kinds of plants. It causes cosmetic damage to strawberries as they mature. So it reduces fresh market yield. And the management approaches to this pest, excuse me, either because of insecticide resistance in conventional fields or other management tools or strategies in organic fields were lacking. So we definitely need to come up with an approach here. This is the lygus bug nymph. This is the immature stage. This is the stage that causes a lot of the feeding damage. So the approach, the innovative approach that we took here was to use trap crops, specifically alfalfa. So trap crops are designed to be highly attractive to a pest, much more attractive than the cash crop, in this case strawberry. And so you pull them away from the strawberry and kind of confine them in this alternative planting. They're attractive, alfalfa is. It retains the pest really well, lygus bug. Excuse me. Lygus bug. It makes scouting and management, in other words, managing the pest really, really efficient because you know where they are. I'll show how it promotes biological control. And it improves yield too, so it's good for the farmer. Again, everything has to improve yield. Always has to improve yield. So here's what it looks like. These are organic strawberries with alfalfa trap crops. Alfalfa has those really nice purple flowers. They're really pretty. And the lygus bugs are attracted to those trap crops and they get aggregated in those spaces. So we worked with some collaborators to look at movement and see how movement could be influenced by the presence of this preferred plant host. This is using protein marking techniques. So you can see the egg whites and the milk. So those were the proteins that were applied to sort of determine how these lygus bugs moved around. We did this with James Hagler. He's sort of a big shot at USDA. He pioneered this method. And let me say, when teachers make you work in groups, it can seem like a pain sometimes, I'm sure. There are differences in personality, differences in how to approach a project, differences in any number of things. The trend in science is to be collaborative. The trend is to work in larger and larger groups. The trend is to be multidisciplinary. The trend is to be multi-state, multi-region, multi, right? The days of these sort of lone wolf researchers and scientists who publish these massive papers with just their single name, that's so rare now. Normally, the journal's trying to figure out how to cross some names off of the author list because there's so many. The road to science is to have strong interdisciplinary or interpersonal skills. In other words, to be a good people person. And if you can do that, it's fun. You get to work with really fascinating, interesting, brilliant people, and it can be really enjoyable. Anyway, you have to be good with people. That's what I'm trying to get at. Okay, so in this case, hopefully if you're from California, this is a scene that is familiar to you. In the spring, we have green hillsides that are in bloom, and then as spring turns to summer, they dry out and become brown. So what ends up happening is the lygus bugs, which are in the green hillsides, they love the flowers, they're there. They're perfectly content in the spring, and then as those hillsides dry out, then they look for green pastures, pardon the pun, and then they fly into the strawberry fields. So we want to test the hypothesis, well, can we prove that as lygus bugs move from the green hillsides or the browning hillsides, will they preferentially fly into an alfalfa trap crop rather than a strawberry field? So here is a protein being applied. It's a reverse, sort of like air blower, but in this case it's been modified to apply protein. I don't know if this is egg or milk, onto some wild radish, which is right next to a trap crop strawberry field. You can see it's a little faint. The contrast isn't great, but you can see the protein being applied. And then we used a modified sort of air blower. This is a vacuum, and there's a net on the end that catches the insects as they get sucked in. And so we captured the insects, and then we did an ELISA. James did the ELISA. Are you guys familiar with this technique? EnzymeLinked Immunosorbent assay. It's fantastic and really versatile and used in all different sorts of things. Essentially, you set up an ELISA to look for a particular protein, in this case egg or milk, so albumin or casein. And it detects the protein, and then it's linked with an enzyme that can turn a particular a bright color. And it's that color that informs you that the protein is present in that particular, in this case, insect sample. And so the dark wells are positives based on this ELISA technique. So here we have the weeds that were marked with milk and egg, or albumin and protein. And we collected lygus bug adults 1 day, 2 days, and 2 weeks after the protein was applied. And you can see the alfalfa trap crops and the strawberries that were adjacent to those weeds and further away from those weeds at the same time frames, so 1 day, 2 days, and 2 weeks after the protein was applied. And you can see that 85% of the adults that we captured flew into an alfalfa trap crop rather than the strawberry, right? In agriculture, and particularly organic agriculture, prevention is the name of the game, right? You really want to prevent your pest from establishing in the field, and if you can do that, you're really buying yourself several weeks of really improved yield. So this was very, very fruitful, again, sorry for the pun, and was a good demonstration of how the protein marking technique can be used in this trap crop environment. Through the same collaboration, we also wanted to characterize the predator community that's found in strawberry. So this had never been done, to look at what predators are present in strawberry and what are they doing. It's a little frustrating, but all these predators are generalists, which is to say that they can eat any number of things, right? They're a little bit flaky in terms of what they eat. This happens to be a damsel bug on the left, and it's conceivable that it would eat a non-pestiferous herbivore, in other words, an aphid, which isn't a pest in the system at the bottom, a lygus bug nymph in the middle, which would be a pest and would be really productive if that predator ate that pest. Or it could eat another damsel bug. It could eat another predator, which is called intra-guild predation, right? You have one predator eating another predator. That's actually counterproductive from the perspective of biological control. So in this case, what we wanted to do was take a look at the guts of these predators and use PCR, polymerase chain reaction. You guys know that technique, right? Okay, so you're amplifying small bits of DNA so that you can verify their presence. And so we wanted to look for lygus bug DNA, the pest DNA, in the guts of the predators to determine whether or not they had eaten, in fact, on that pest, to figure out, okay, how useful is this critter in improving yields. So we collected the predators from alfalfa and strawberry using the same device. And don't worry about the details, but just know that lots and lots of these predators and spiders. Gosh, if any of you want to go into a field that is underrepresented and where we know very, very little, spiders, we know nothing about spiders. Turns out tons of spiders are fantastic predators on this pest. And so we have to do a lot of work to try and figure out what the heck they're doing in this field. The gray bars are the pest. And you can see the pest in TC, the trap crop. So in other words, where you have alfalfa, you have lots of pests. And then in the adjacent strawberries, you don't have that many pests. The red line is the percentage of the predators that we collected that had consumed a lygus bug, that had consumed a pest, right? Does that make sense? So the percentage of predators that consumed a pest. And you can see that the percentage of predators that consumed a pest was highest where there were the most pests, which is in the alfalfa. And that's called conservation biological control, right? You're able to conserve or improve or augment the biological control that's being present, in this case by jacking up the prey availability, but in an area that doesn't compromise your yields. That's the tricky part. That's where most people have a hard time. So we're able to do this in alfalfa, which doesn't compromise your yields, and it improves predator performance. Okay, here is a parasitoid. This is Parastinus relictus. And does anyone have any idea what the difference is between a parasitoid and a parasite? Any guesses? Okay, does a parasite intend on killing its host? Is that part of the plan? No. Sometimes it happens inadvertently, right? But that's not the plan. A parasitoid, it's part of the plan to kill its host. And this little wasp, it's about the size of an ant. That's its job, is to kill its host. So here's a female wasp, a female parasitoid, and she's using her stinger, ovipositor, to insert an egg inside of a living lygus bug nymph. And that egg is going to hatch out into a little larvae, and that little larvae is going to eat the inside of the lygus bug nymph while still alive. So this is just an adult parasitoid. And you can see they're not very big, they're really small. They do look like ants with wings, right? Feeding on sugar here. And here's Charlie Pickett over at CDFA. He really spearheaded this project. He's another great person, another awesome collaborator that we've been able to work with. So we collect the nymphs, and then we dissect them. And what you can see is the larvae that's coming out of the dorsal side of the nymph, the back of the nymph, right? So that's the larvae that is mature, that had eaten the inside of the nymph. And it's incredible sometimes. You can see these nymphs, their whole interior cavity is no longer occupied by organs, but occupied by these larvae, right? They're just occupying the whole interior cavity. And the nymphs are still amazingly walking around. They're still alive. They're kind of like zombies, right? There's nothing really going on metabolically, but it's incredible. So here's another larvae that's coming off the posterior end of the nymph. All right, so this is a little complicated. If you look at the x-axis down at the bottom, right, there's the trap crops, the TC. The nymphs, the pest, is on the first or the left-hand side y-axis, and the number of larvae, or the number of parasitized nymphs, if you want to look at it that way, is on the second y-axis or the right-hand side y-axis. Basically, there's a tight correlation between how many pests there are and how many pests get parasitized, and that happens in alfalfa, right? Again, similar to how many prey there are and how many prey get eaten, here we have how many hosts there are and how many hosts get parasitized, and in both cases that happens more often in alfalfa than it does in the strawberry. This is the slide where growers all of a sudden get on the edge of their seat and they start paying attention. They may or may not be paying attention earlier, but for this slide they definitely pay attention. So on the top histogram, you can see the percent damage, in other words, the percent of fruit that are damaged by lygus bug. The trap crop strawberries are the light gray, the control strawberries, the untreated control strawberries are in the dark gray. And so you can see that in 4 years, 3 of the 4 years, you got reductions, significant reductions in the percent of fruit that was damaged. If you want to look at it the other way, on the lower histogram, if you look at the mean number of good fruit, harvestable fruit per plant, right, that's higher in the trap cropped plants. And that could be anywhere from a third to over a half of fruit, a berry per plant per harvest date. And we're talking about 20,000 plants per acre. Right? This is a lot of improved fruit that growers are able to pick. This is very significant in economic terms. Okay, let's transition a little bit. Let's look at olives, and in particular olives at Kenyatta College. Are olives native to California? No, right? Have they been here a long time? They've been here a long time, right? So the Spanish settlers that came over and set up the missions, they brought olives. They were the first, the ones to introduce olive trees to California. So back in the late 18th century is when olive trees were first introduced to California. Do we kind of consider them to be Californian, sort of the California landscape? Yeah, right? There are lots and lots of plants that we consider to be part of the California landscape that aren't technically California. And this is for good reason, at least in the case of olive, right? So almost all of the olive oil is produced in the country, is produced in California. I just got this today on the California Olive Oil Council website. 75 different olive varieties grown in California. I didn't know that, 75. All with different flavor profiles. And just as a little side note, if you guys are at all interested in olive oil, you definitely should check out the California Olive Oil Certified Extra Virgin label that they have on the bottles. It certifies that it's done right and it's going to taste the best and be the healthiest for you. A lot of the olive oil that gets imported over from Europe, it says it's extra virgin, but it's really not. It's kind of garbage. So a good way to verify and guarantee that you're getting high quality olive oil, it's really good for you, it tastes really good, is to buy California with that label on it. Okay, at any rate, here's the logo for Kenyatta College. What's the logo? Olive, an olive tree. Do we have lots of olive trees on campus? Yes, so many of my students, I say, you don't have olive trees. I go, where? I go, there are hundreds of olive trees on campus, right? You can't take 10 steps on campus without hitting an olive tree. They're all over the place. This is what olives should look like, and at least potentially can look like. But without intervention, this is what olives do look like, right? So big difference. And the olives look like that because of the presence of the yellow fruit fly. This is an exotic insect. It is present just about everywhere olives are grown. For some lucky reason, California had avoided this pest for a couple centuries. Right, so we've had olives in California since the late 18th century. It only first got to California a few years ago, so we kind of lucked out for a long time, but our luck finally ran out when we got the fruit fly. So there's a female fruit fly. It goes from the top left box across. She'll oviposit or lay an egg inside of an olive, and you can see the sting marks. So when olives are not quite ripe, when they're ripening, you'll see these little dots on them. Those are the oviposition marks where fruit flies lay an egg inside. And what do flies lay eggs? And what they turn into, we've already established, maggots, right? In this case, these maggots are bad, they're not good. So the maggots are feeding on the inside of the ripening olive, and they'll tunnel all the way through. You can see it on the bottom left-hand corner of the slide. And eventually, they end up emerging, and as soon as they emerge, then they allow bacteria to enter into the olive, and that causes rot. And then they'll emerge and they'll pupate. Pupate means pupa, which means cocoon or chrysalis, right? Sort of the stage in between the larvae and the adult. Yeah? A codling moth. Yeah, apples have codling moth. It's the same basic process. In this case, it's a moth, which is laying an egg, and it's a caterpillar, which is burrowing through the apple. Same process. So here is the temporal and spatial distribution of the first reportings of the olive fruit fly in California. So it first arrived in LA and Orange County back in the late 90s, and it only took a few years for it to make it up to San Mateo County in 2001. And it's now pretty much all up and down the state. Anywhere olives are, you'll find this pest. And that's the thing about new pests, exotic pests. If they're mobile, and if the weather is accommodating, they can disperse very quickly. All right. There are centers of plant domestication, centers of plant diversity, sort of these hotspots where a lot of plants ended up being domesticated to produce the foods that we eat. My favorite happens to be in Mexico. Avocado, beans, chocolate, chiles, corn, pumpkin, squash, tomato, cotton, vanilla. That's not even including tequila. So there's lots of good stuff that comes out of Mexico, right? A lot of good stuff. Do these crops end up staying where they have evolved? No, right? Of course not. They move all over the place. With globalization, you end up growing these crops all over the place. California, we're also a citrus state that comes from Southeast Asia. A lot of the crops that we consider to be ours, quote unquote, don't come from California. So you guys all know the basic food pyramid, right? You've got your producers at the bottom, you have your primary consumers or your herbivores next, and then your consumers, your predators up top. So when we move crops from one location to another, right? Very often, we're not just moving that crop, we're moving the herbivores, the pests along with it. Excuse me, and we're not trying to, but they're really good at hitchhiking. A lot of them are really good at sort of just grabbing on, holding on, and before you know it, you have kind of half a system, right? You have the bottom half of this sort of trophic food web, where you have the pest, but nothing to control it. So what do we control it with? Yeah, we control it with chemicals, right? Which is fine in the short term, but as we've shown, there are some consequences that are not exactly favorable. What classical biological control tries to do is it tries to complete that pyramid, right? It tries to reestablish the missing trophic levels to try and bring some ecological balance to a system. And in the case of olive, so we now have thousands of miles of olive in California, right? So olive trees they're on campuses, they're in hillsides, they're in homes, they're in parking lots, they're in orchards. The whole state is just all coast, inland, desert, mountain, right? We've got olives everywhere. So you now have this breeding ground for this pest without any natural controls. So the process of classical BC is to try and rectify that situation. So foreign collections are the first step. In other words, you have to go to where the pest originally evolved and find the beneficial predators and parasitoids that co-evolved with that pest. Any guesses as to where olive fruit fly evolved? Yeah, not Italy. A lot of people think that's a really common, it's a really good guess, right? But yeah. So it turns out sub-Saharan Africa is everyone's best guess, which is where this thing evolved. And you can see wild olives, they're super small, and they're produced year-round. And that's where this thing evolved. And I'm sort of excited to say, we just, this last year, had roughly 6 wild olive trees planted on campus. So they're not fruiting yet, but hopefully they will in a year or 2 or 3. And we'll be able to have some wild olive trees on campus. So you do some foreign explorations at the point where the pest originally evolved, and you collect the good bugs. You collect all the good bugs you can, and you put them in quarantine. In this case, the quarantine is in a USDA laboratory in Montpellier, France. And so here are some of the different candidates that were being tested in quarantine to figure out their appropriateness or their eligibility for release in California. So what happens is you figure out, okay, how specialized are these parasitoids on the pests that we're looking for, right? In other words, if you expose them to non-target hosts, things that are similar kinds of insects that are also found in California, are they likely to go after those non-target insects in California? Or are they going to be specialized and limited to only the pest in question, the target pest, in this case, the olive fruit fly? So after those tests were done, and they're really very long, very involved, very expensive, lots of permits, lots of regulations. There's a lot of very good, long, responsible work that needs to be done. This isn't willy-nilly. People don't take this lightly. There were 2 candidates that were identified, Cytalia humilis and Cytalia lounsburyi. And humilis is taken from Namibia in South Africa, lounsburyi from Kenya in South Africa. Here's a picture of a lounsburyi. It's got really cool long antennae. And there's sort of a Rorschach test on the dorsal side of its thorax. Everyone kind of sees a different image there. And then here's humilis. It's a sort of a really pretty bright amber color, again with long antennae. And so here are the release sites. This is a massive project, right? So CDFA, USDA, the University of California, were all involved to try and implement a statewide response to this infestation. So anywhere you see a highlighted county, that's where releases are taking place. The strategy for this biocontrol is to use urban centers that are adjacent to ag centers. The thinking being that the urban centers are not going to be sprayed with insecticides, and so you have a better likelihood of establishment. So here's Charlie Pickett again. He's on campus. Here's Xin-King Wang. He's a researcher from UC Berkeley. And here are the parasitoids in the vials. They're being released here on campus. They're really cute. Here they are in the olive. Okay, so the results. I think the most important thing here is that Cytalia lounsburyi was first captured in 2011, and Kenyatta College was the first place in the state where this parasitoid was captured. It was a really big deal, and it's been captured every year since. So we have a definite establishment that seems to be happening with this particular parasitoid, using Kenyatta College as sort of the epicenter for that establishment, at least in Northern California. This last particular year, my students and I did some pretty extensive collecting up and down the peninsula. You can see the distance from Kenyatta College and the number of olives that were collected in each of these different locales. Well, if you look at the bottom, San Francisco and San Jose, you can see in the P. lounsburyi column, you've got parasitoids that were picked up both in San Francisco and San Jose, which was amazingly exciting. So here we are, Kenyatta College. It served its primary purpose, which was to act as a source of incubation for the peninsula to act as that primer for establishment. And so we have now the whole peninsula which has these parasitoids. It's really, really exciting. And these results were published in a peer-reviewed journal in biocontrol, and you have these authors from UC Berkeley, UC Riverside, USDA, CDFA, and Kenyatta College. So next time you guys are walking under an olive tree on campus, there could be some interesting things that are happening just sort of out of sight. Okay, so I was told I was supposed to give some advice. At least with respect to how my career has gone, I would say follow your passion. Folks are working really hard these days, right? Your work never kind of seems to end. And I can't imagine working that hard on something that I didn't enjoy, didn't feel passionately about. So my first bit of advice, follow your passion. Be proactive, particularly with internships and with interactions with faculty. You really have to, faculty are so busy, right? So the more proactive you can be to volunteer, to try and get your foot in the door, the better off you're going to be, right? That is so important. Look into some of the educational requirements and professional norms that are present for something you might have in mind. Develop strong interpersonal skills, like I mentioned. No one wants to work with a jerk, right? And I'm telling you, in these multi-agency collaborations, if you have the choice between, one guy who's a nice guy and someone else who's a jerk, and they both do the same things, who are you going to go with? You're going to go with the nice guy. And so in STEM, right, you're always told math, 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 and it's definitely true, but I say write. I think it's critical that you are good writers. If you want to get a grant proposal funded, it has to be well written. If you want to get a manuscript published, it has to be well written. If you want someone to collaborate with you, and you write an email, and they don't know you, the only thing they know is how well that email's written. Your success is dependent upon how well you write. I'm telling you, if you're reliant on grants, literally you're writing for your life. And I feel like math gets emphasized, which is fine, but you can always hire a statistician to help you out. You can't hire someone to write a grant for you. You have to be good writers. Okay, and with that, are there any questions? Yes? Sure, so the question is, what's the need to be more collaborative in your projects? Part of it is everyone's very specialized in their research discipline, and so if you want to address a broader, bigger question, you need a specialist in various fields to answer that question. The other part of it is there are fewer grant dollars around, and so you have more people going for fewer dollars, and it's increasingly competitive to get those grant dollars. And so if you team up with more people who have success, you're more likely to get those grants. Yes?
Could you talk a little bit more about what you have to go through to ensure that these parasitoids wouldn't affect the ecology, and because you have to do experiments, you have to, whatever regulatory bodies are in the county or state.
Sure. The question is about guaranteeing the safety of parasitoids before you make releases. USDA APHIS is very strict about guaranteeing the safety of non-target hosts. Years and years of quarantine testing has to be done. So in other words, choice and non-choice tests with the prospective insects that are going to be possibly affected in that environment. Also, a lot of stuff has been done before, so you can rely on previous studies, other work, and it's just a big effort. The way the regulatory landscape is shaping up, if there are any doubts about the safety of a release, you won't get permitted. So the agencies are definitely erring on the side of caution, for sure. Yes?
Do you think is there a possibility that in the future there could be some kind of genetically engineered insects or parasitoids that could make human their host and then turn them into zombies or spider?
Wow. Good question. So there are genetically engineered insects. We've already gotten to that point. They tend to be engineered not to carry malaria, things like that, with vectors, mosquito vectors. That's been the the forefront of genetic engineering for insects, so we're there. We're definitely there. The zombie part. There are some insects that are parasitoids, like for caterpillars. Caterpillar parasitoids, they kind of make zombies out of their host. They already do that. They take neurological control over the host and they have the caterpillar that's already essentially dead kind of defend the parasitoids while it's still alive. So at least sort of in a sci-fi reality, sure, that's not that big of a leap. It's not that big. So we're already genetically engineering insects, and insects are already using their host as zombies, so I don't see why not. Sure. Any other questions?
Is there one more question? Yeah.
What can one do to reduce the amount of pesticides one consumes?
Good question. Other than buying organic, right? But if you only consume conventionally managed or produced food because it's less expensive, the presence of a peel is really important. So citrus, banana, pineapple. If you can remove the peel, that is going to carry the burden of the pesticide residue, and so that's not what you consume. Compare that to a peach, strawberry, a cherry. There's no barrier, there's nothing that can be removed that stands in the way between you and the spray rig. And so the environmental working group always has their dirty dozen list every single year, and they, based on USDA data, they list the produce items that have the highest pesticide residues. And so you can check that out. Yeah. Okay, thank you.